soon. There we go. All right. So today it's chapters 3 and 4. And we'll talk a little bit also about Connect and what's going on there, if you're having any trouble. Okay. So, good. Okay. What we want to talk about in chapter 3 is the anatomy and physiology of the microbes that you're going to be studying during the course of these 10 weeks. Okay. So we're going to talk a little bit about what a prokaryotic and a eukaryotic cell are, what sort of machinery they have, how they work, how the parts operate together to sustain the cell. And by doing that, we're going to understand enough about these organisms that we'll also be able to determine how best to attack the ones that cause disease, those that are what we refer to as the pathogens. Okay, so we'll talk about the different bacterial shapes, the structures. We begin with the bacteria and the archaea. Okay, now these are prokaryotes. All right, let's write that down and understand what that word means. Prokaryotes. Okay. Okay. These are organisms without a true nucleus. Okay. Pro means before, and karyo means nucleus. Okay. So they're different from the kind of cells that you and I are made out of. The cells are less complex. The DNA is packaged without proteins that are called histones. There's no membrane around the nucleus. The makeup of the cell wall is a unique structure in prokaryotes. In bacteria, it's made of something called peptidoglycan. There are other polymers that make up the cell walls of archaea. Okay. Our cells don't have cell walls. Okay. Animal cells lack cell walls, and plant cell walls are made up of something called cellulose, which you know better as wood. Okay. So those are unique characteristics. And the reason we're interested in unique characteristics is because they help us to identify something so that we are able to understand the rest of its characteristics and how to deal with it. Okay. There are no membrane-bound organelles in bacteria and archaea. Okay. There are non-membranous organelles. There are ribosomes. There are scaffolding made out of protein. Okay. There are basal bodies to which flagella attach. Okay. But none of those have membrane around them. The only thing that is surrounded by a membrane in a bacteria and archaea is the cytoplasm. Okay. Okay. All bacterial cells have a cell membrane. They have cytoplasm. They have ribosomes. They have at least one chromosome, and most have a cell wall and a surface coating known as a glycocalyx. Okay. So let's put a little detail in about these. Cell membrane is made out of phospholipid, and proteins. Okay. Some of the proteins go all the way through the phospholipid bilayer. Some of them are on one side or the other. Okay. The cytoplasm is a gel-like semi-solid with proteins electrolytes, water, okay, nutrients, all sort of embedded in the semi-solid. Ribosomes are protein builders, protein building organelles. Okay. The cytoskeleton is a protein network that supports the cell. Okay. The chromosomes are where the genetic information is located. And when we say genetic information, we mean protein building information. Okay. The cell wall is a hard outer layer of peptidoglycan, okay, and the glycocalyx is basically a sugar layer. Okay. Some, but not all, bacterial cells have flagella, pili, and fimbriae. Okay, flagella are for locomotion. Flagella. Okay. Pili are for mating. We'll get into that a little bit later. Okay, and fimbriae are for attachment. They have an outer membrane. They have plasmids. What are these? This is extra chromosomal DNA. Okay. They have inclusions. These are chambers for storage. Okay, and we say storage of what? Pigment, waste products, depends on the cell. Endospores, which are sort of like seeds. Okay, it's a form that the bacterial cell can take on when environmental conditions are poor and rapid growth is unlikely. And endospores are almost impossible to destroy and almost immortal. Okay, have intracellular membranes in some cases. Okay, and what you're looking at here is just a diagram showing you a lot of these different structures. I won't go through each one, but this figure is in your text. Okay, so it sort of outlines what each thing is, just like we were talking about before. Okay, so it's a very simple cell. Okay, most bacteria function as independent single-celled unicellular organisms, but some can be groups in the context of what's called a biofilm or as a colony. Okay, so a biofilm is many bacterial species working together, right, in a sort of a matrix. And a colony is a single bacterial species. Okay. Some communicate through nanowires. That's usually seen in biofilms. They're very tiny. They're a micron large. Okay. And so that means that on a light microscope, you're going to have to use the oil immersion objective in order to see them. And they're going to have to be stained. Okay. So very, very tiny. Cocci have a circumference around a micrometer, rods between 2 and 1 micrometers. And some have a multiple series of shapes, and they're called pleomorphic. Okay. And that can be the result of within a single species differences in the genes they carry or differences in their nutritional status. Okay. Now, in order to see bacteria, we have to stain it, or else it'll be transparent, right? So one of the things we do is that we can spread some of the bacteria on a glass slide, we can heat fix it, and then we can hit it with a stain and look at the different shapes and the different arrangements. Okay, so let's go through a staining procedure here.
Prokaryotic microorganisms exist in a variety of different cell shapes. Some are spherical, but seldom are seen as a single cell. A spherical cell is called a coccus. Pairs of spherical cells are called diplococcus, the plural being diplococci. Chains of cells are streptococci, and grape-like clusters of cells are staphylococci. Some genera, such as Streptococcus and Staphylococcus, derive their name from the most common pattern of arrangement of the cells. Many prokaryotes are rod-shaped in appearance. A rod-shaped cell is called a bacillus. The genus Escherichia consists of rod-shaped cells. The genera Bacillus and Clostridium consist of spore-forming rods. Spore formation in some bacteria causes swelling of the mother cell. There are also many other genera of rod-shaped prokaryotes. Bacilli occasionally occur as chains of cells called streptobacilli. Cells of the Corynebacterium group have a characteristic club-shaped appearance. Some bacteria have a curved rod or vibrioid appearance. An example is the genus Vibrio. Mycoplasmas are a group of bacteria that lack a cell wall. The cells of this group are pleomorphic, meaning that the cells take on a variety of different shapes. There are two groups of spiral-shaped bacteria. Spirilla are motile by means of flagella, and spirochetes are motile by means of axial fibrils located beneath a sheath that covers the entire surface of the cell. Bacteria such as the Nocardiae form branched filamentous cells. These cells reproduce by fragmentation. Actinomycetes form branched filamentous cells. This group is characterized by formation of a variety of specialized dispersal cells that form on the aerial mycelia. In some species, these cells consist of strings of individual small cells, whereas in other species, the dispersal cells are found in sacs called sporangia.
When we look at bacterial sizes and shapes and arrangements, we have three basic shapes, right? We have the cocci, which are round like BBs. We have the bacillus, which are rod-shaped, sort of like a Tylenol capsule. And then we have spirochetes, which look like springs. And then we have those that are sort of multiple shapes, the pleomorphic ones, or the ones that lack a cell wall. The arrangements can either be in chains or in pairs or in tetrads or in clusters, okay? If it's in pairs, they are called diplo, and then the shape follows. If it's in chains, it's called strepto, and then the name follows. If it's in a tetrad, okay, we can refer to them as sarcina, okay? And then if it's in several planes, division look like clusters of grapes, we call those staphylo, okay? So you can have diplococci, streptococci, sarcinococci, and staphylococci, and the same for bacillus, and even sometimes for spirochete. Okay. And you might wonder, well, okay, why do we care? We care because this tells us something about where the cell division takes place, and that leads to the specific arrangement of the cells. And that's a characteristic that's going to be unique to certain bacteria, right? So here you divide in one plane, right? Here you divide in two perpendicular planes. Here you divide in several planes, and so on, okay? And that can help us to characterize the bacteria. Okay, so that's why we care. Okay, cocci can be single, double, tetrad, staphylo, strepto, right? Bacilli, single, diplo, strepto, or sometimes palisades, where it looks kind of like a chandelier. Okay, or spirilla, which are found in short chains. Those are like spirochetes. They look like springs. Okay, and spirochetes, which are like individual springs. Okay, so all different possibilities, right? You got the round ones, you've got the rod shaped ones, spirochete shaped ones, and you can arrange those in a variety of different ways. And then some are vibrio, some are pleomorphic, meaning they have several possible shapes. Okay. Um, so all those are possibilities out there. Okay. Okay. Now, there are going to be external structures on some of these bacteria that help them perform certain tasks. All right. Appendages that are used for motility include flagella and axial filaments. Okay. An axial filament is embedded. Okay. Whereas a flagella sticks out. Okay. Fimbriae and pili. Okay. Fimbriae are for attachment. Think Velcro. Okay. And pili are for mating. You might say, what do you mean mating? Well, what we mean really is DNA transfer. Okay. So they can get together and they can grow this hollow pipe between them, and they can duplicate their DNA, and they can slide that DNA over to the recipient. And then the recipient gets those pieces of information. Flagella is for motility. The three parts are the filament, the hook, and the basal body. Okay. You can see the parts here, right? There's the basal body, right? There's the filament, and there is the hook. Okay. And you can see here, it spans three outer layers, right? It spans the cell membrane, the cell wall, and the outer membrane, which you see in gram negatives. Okay. So let's indicate that they're gram negatives. What do I mean by gram negative? I mean that in a gram stain, it's going to end up looking pink. Okay. All right. Now, where does the power come from? The power comes from ATP. And what this thing does is it spins around and spins around as it burns ATP, and it can either spin clockwise or counterclockwise. All right. We'll learn a little bit more about that in a bit. Okay. And that's an actual micrograph of a flagella. Okay. I had to take that with an electron microscope. Okay. Flagella can have different arrangements. They can have a polar arrangement where there's a single flagella at one end. They can be lophotrichous with tufts of flagella coming from the same site, sort of like a horse's tail. Okay. They can be amphitrachous, which means they can be at both poles of the cell. And they can be peritrichous, which means they're all around the cell, right? So again, the prefixes tell you, right? Mono means one, right? Lopho means tuft, okay? And amphi means between, okay? So the amphitrichous condition is between monotrichous and peritrichous, okay? That's what it looks like, right? There's monotrichous. There's Lopho. Okay. Amphi. And there's peritrichous. Okay. So different arrangements, right? But in every arrangement, these cells can move in any direction. Okay. How do they do this? They do this using something called chemotaxis, right? Which is the ability to run away or towards a set of chemical conditions. Okay. In positive chemotaxis, you move towards a particular chemical gradient from least concentrated to most concentrated. In negative chemotaxis, you run away from it. Okay. The run results from a counterclockwise spinning of the flagella so that it zips in a straight line, whereas a tumble is a clockwise rotation of the flagella, and that cell then stops and changes its direction, and then you can have a run in the other direction. Okay, so what's driving this is a series of chemical reactions that take place inside the cell. All right. So what follows here is just a discussion of the mechanism whereby the flagella work. What I'm more interested in you understanding from this is what a run and a tumble are. You know, what is a run versus a tumble? Okay. Okay. And then what is positive versus negative chemotaxis? Okay. Those are the most important takeaways. And this video will get into a little bit more of the mechanisms whereby that works, which are interesting, but you don't need to memorize those details. I just want you to know those main points up top. So let's take a listen.
When Escherichia coli moves in a medium that lacks a concentration gradient, the cell travels, stops, or tumbles, and then continues moving in a new random direction. When the flagella rotate counterclockwise, the flagella form a tight bundle and propel the cell forward in a run. After a brief period, the direction of rotation is reversed, causing a tumble. As cells move up a chemical gradient, the runs are longer than when they travel down the gradient. The overall result is random movement in the absence of a chemical gradient and movement toward a chemical when a gradient exists. Attractant chemicals bind to chemoreceptors called methyl-accepting chemotaxis proteins, located in the cell membrane. The cytoplasmic side of the MCP interacts with two proteins, KW and Ka. When the MCP is not bound to an attractant, it stimulates Ka to phosphorylate itself using ATP. Ka autophosphorylation is inhibited when the attractant is bound to its MCP. Phosphorylated Ka can donate its phosphate to the response protein Ky. When Ky is phosphorylated by Ka, it moves to the flagellum. This causes the flagellum to rotate clockwise and tumble. Thus, a decrease in attractant promotes short runs and tumbling. KW is an adapter protein, mediating communication between receptors and Ka. The phosphate on Ky is continually removed by Kz. The short lifetime of phosphorylated Ky means that the bacterium is very responsive to changes in attractant concentration. When no attractant is present, the cell maintains intermediate levels of key A phosphate and key Y phosphate. This produces a normal run-tumble swimming pattern. When a chemoattractant binds to MCP, the levels of key Y phosphate drop. The lack of phosphorylated key Y results in counterclockwise rotation of the flagella and long, smooth runs. Escherichia coli must be able to ignore past stimulus responses so that it can compare the most recent attractant concentration to the immediately previous one. This is accomplished by methylation of the methyl-accepting protein. Key R continuously methylates MCP, whereas phosphorylated key B removes these methyl groups. When the attractant is bound to MCP, the complex becomes a good substrate for key R and a poor substrate for key BP. The levels of key YP and key BP drop because phosphorylation of key A is inhibited. Removal of the attractant causes the overmethylated MCP to stimulate key A autophosphorylation, and the levels of key YP and key BP increase. This simultaneously induces tumbling and demethylation so that the system returns to a lower level of key A autophosphorylation.
Now, periplasmic flagella are something that are seen in spirochetes, which are shaped like little springs, and they spin, okay, either clockwise or counterclockwise because of these flagella that are in what's known as the periplasmic space, which is between the cell membrane and the cell wall, okay? They're also known as axial filaments, and they allow these springs to twirl on their long axis and move either towards or away from a chemical gradient. So here's a look at how the periplasmic flagella operate.
Most motile bacteria move by means of flagella. A flagellum is a slender structure about 15 to 20 microns long and about 20 nanometers wide. A typical rod-shaped bacterial cell is 2 to 3 microns in length. The bacterial flagellum is composed of 3 parts: the basal body, which is embedded in the cell membrane and wall, a short curved segment called the hook, and the portion that extends out from the cell called the filament. The basal body consists of rings that correspond to the layers of the cell envelope and therefore differ in gram-negative and gram-positive bacteria. In most gram-negative bacteria, there are 4 rings connected to a central rod. Gram-positive bacteria have only 2 basal body rings, one connected to the cell membrane and the other to the cell wall. Motility is mediated by proteins associated with the plasma membrane and basal body, and is powered by the proton motive force associated with the plasma membrane. The filament is a rigid structure which rotates to move the bacterial cell. Spirochetes are motile by means of bundles of flagella, called axial fibrils, that run along the cell within the periplasmic space. The number of fibrils varies. These axial fibrils extend from both ends of the cell within a flexible outer sheath. Spirochetes have a screw-like motion which allows them to bore into mucoid layers and soft agar media. Some bacteria lack flagella or axial fibrils, but are motile if they are in contact with a solid surface. This type of movement is referred to as gliding motility. For some gliding bacteria, secretion of a slimy polysaccharide appears to be involved. Other bacteria appear to have special proteins on their surfaces that mediate gliding motility.
Now, fimbriae are basically for attachment. I don't know if you've ever had something that seals with Velcro, but that's basically what this is like. They allow the bacteria to cling to a surface and then grow on that surface and colonize it, okay? The pili are hollow tubes through which genetic material can be passed, okay? Gram-negative bacteria use pili to transfer genetic material from one cell to the next. The advantage of this, of course, is the ability to pass on specific traits from one cell to another, such as drug resistance, okay, or what we call virulence factors, which are a lot like weapons, okay? So you can see an example of pili and fimbriae here, right? Fimbriae are basically the projections that are surrounding the cells sticking out, right? The dark sort of halo around the cells. And then you could see the pili running from one cell to the next, okay? So that's one of the reasons that bacteria can acquire drug resistance so quickly. If one of the cells in the bacterial population has a piece of DNA on a plasmid that allows it drug resistance, it can duplicate that plasmid and send it to another cell, even another species, right? And transfer those characteristics to a new cell. And now that cell comes away with the resistance as well, okay? The S or slime layer are basically proteins that are linked together, sort of like chainmail around the outside of the bacterial cell. And they're generally produced when the bacteria are in a hostile environment. So think of it like body armor, okay? Well, the glycocalyx is a polysaccharide layer. Sometimes there's glycoprotein that aids in things like attachment and recognition, right? Sometimes there is a looser layer called the slime layer that can protect against loss of water and nutrients, and can also provide a degree of protection against immunity for the bacterial cell, because it makes it difficult for the white blood cell, say, to grip the slime layer. And the capsule is tightly bound and denser and produces mucoid characteristics on agar, and can also provide protection against immunity. Okay, so the slime layer and the capsule are kind of like armor against attack. Okay, so you can see some examples of that here, right? You can see the glycocalyx, and this would be the order in which the layers would occur if a single bacterial cell had all of these, right? You'd have glycocalyx on the very outside, you'd have the S layer beneath that, then there's the cell wall, and then there's the cell membrane, okay? So look at all those layers, right? You got 1, 2, 3, 4 layers, 4 layers, right? All have different characteristics. And what does that mean? Makes it more difficult, makes it tough for drugs to penetrate, or for immunity to work, right? Guards the cell, right? You could see some examples here, right? Note the, again, this is a capsule stain, negative stain, but note the light area around there showing the capsule, right? This is a red bacteria called Serratia sessions. Okay, red colonies. What else can the glycocalyx do? Well, the capsules, which are used by pathogens, allow an increased ability to produce disease because they can protect against phagocytosis. Now, if you don't remember what phagocytosis is, that is endocytosis of solids. It's a form of active transport, and this is carried out by white blood cells. Okay. Biofilms are communities of bacteria that grow within a matrix that allow it greater resistance to attack because of the variety of bacteria that live there, right? So you've got many species. Many species of bacteria with many weapons, okay, different weapons to protect against attack. It's responsible for colonization of catheters and IUDs, pacemakers, and indwelling devices, right? Big problem, especially in hospitals, okay? So let's look a little bit deeper at the glycocalyx.
Clean abiotic surfaces immersed in liquid tend to attract and concentrate nutrients, whereas biotic surfaces such as plant or animal tissue can themselves be a source of nutrients for microorganisms. A biofilm is formed when microorganisms attach to and grow on a variety of biotic and abiotic surfaces. The initial attachment is usually reversible and often involves attachment by flagella, fimbriae, or other cell surface structures. This is followed by growth of the microorganism and production of an exopolymer, which makes the attachment irreversible. Growth within the biofilm is not uniform. Channels develop which permit incorporation of fresh nutrients and oxygen. Metabolic activities of microorganisms in a biofilm can create changes in the microenvironment. For example, an oxygen gradient can develop that permits anaerobic metabolism near the interior and aerobic metabolism on the exterior. Nutrient and pH gradients are also produced. In the natural environment, biofilms often consist of different types of organisms that function together in the cycling of the elements. For example, breakdown of cellulose on the surface of a decaying plant may result in release of glucose and other carbohydrates, which supports the growth of non-cellulose degraders. Carbohydrates are converted to organic acids and methane by anaerobic bacteria, and the methane can then be degraded by aerobic microorganisms.
So you can see here how a biofilm progresses, okay? You start out with these pioneer cells that generate an organic coating, okay? And that allows other cells to come and stick. And then you get this very sort of complex arrangement of different cell types, which can communicate through what are called nanowires. And that allows them to activate certain genes that generate special products that can help the community do things like get nutrients and defend against attack and build more matrix and all sorts of stuff that help it to be more persistent and more difficult to wipe out. And it's thought that these were the precursors to the first tissues in evolution. Okay, these are loose associations of different cells. As you will know, a tissue is a very intimate association of similar cells that do something as a team that a single cell can't do on its own. Okay, so this is probably the first step in the movement from independently living single-celled organisms to a multicellular body plan. Okay. All right. Next thing we want to talk about is the envelope that surrounds bacteria. Okay, this is outside the cytoplasm and is made up of the cell wall, and beneath that is the cytoplasmic membrane. And then in gram-negative bacteria, there is an outer membrane. Okay, but all the cells have the cell membrane and the cell wall. All right. Now, when we come back from break, one of the things you're going to get an appreciation for is the gram-positive and the gram-negative cell wall. Okay, there's the gram-positive cell wall on the left, all right, and the gram-negative cell wall is there on the right. Okay, so gram-positive, gram-negative, all right, and what do gram-positive and gram-negative mean? They just mean they stain different colors when they're subjected to something called the gram stain procedure, which we'll get into a little bit later. All right. Gram-positive bacteria, as you can see, are purple after the staining procedure is over, and gram-negative bacteria are pink after the staining procedure is over. Okay. But notice that there's always a cytoplasmic membrane, right? There's always a cytoplasmic membrane, no matter what, right? So that's in both of them. There's always a periplasmic space, which is between the cell membrane and the next layer. That's here and here, okay, for both cell, the gram-negative and gram-positive. And then the peptidoglycan layer in gram-positives is very thick, right? Whereas the gram-negatives, it's very thin. And then this extra membrane is only seen in the gram-negative bacteria. So that is a unique feature of gram-negatives, okay? Basically, the more layers you have, the tougher you are to kill, right? Because it's going to be, number one, it's more distance to go, but number two, it's different chemical characteristics, right? And with different chemical characteristics, to get through every layer, you're going to have to be very special, right? So we will deal with this when we come back from break in 15 minutes, okay? So I will see everybody at 2 o'clock, all right? Okay, we had talked earlier about the cell wall, all right? And what it does initially, right, is provide shade for the bacteria. And it also protects against osmotic shock. We know that when a cell enters a hypotonic environment, water tends to rush in and the cell will swell up. And if there's no cell wall, it'll rupture, right? So the cell wall keeps that from happening. That's job number one, all right? But job number two is to protect against attack, all right? There are drugs that can attack the assembly of the cell wall, antimicrobials that damage the production of the cell wall, and that can cause it to lyse under hypotonic conditions, okay? You can see an example of the cell wall structure here. Peptidoglycan is a unique molecule. It is part protein and part polysaccharide. And essentially, the way it's put together, right, you have these unique stretches of amino acids, right? And some of these amino acids are really unusual because they are D amino acids as opposed to L amino acids, right? And that's not something you normally see in a protein, all right? It just means that the chemical groups attached to that middle carbon are arranged in a different way than they are in the L version of the amino acid, okay? And then you using that to cross-link these N-acetyl glutamic acid and N-acetylmuramic acid polymers, right? So it's double tough, right? You've got longitudinal and you've got horizontal stability here, okay? So let's look a little bit deeper at the structure of the cell wall.
Most bacterial cell walls contain peptidoglycan. This structure is essential for maintaining the structural integrity of the cell. Peptidoglycan is a structure that is unique to bacterial cells. This combination of essential and unique makes peptidoglycan an excellent target for antibiotics. The precursor subunit of peptidoglycan, UDP-N acetylmuramic acid, is synthesized in the cytoplasm. A lactyl group is added to carbon number 3 of UDP-N acetylglucosamine to form UDP-N acetylmuramic acid. 5 amino acids are added sequentially to UDP-NAM to form a pentapeptide side chain. Special adding enzymes are used to add these amino acids so that tRNA and ribosomes are not involved. In the case of Escherichia coli, L-alanine is added first, then D-glutamic acid. then mesodiaminopomelic acid. Finally, the dipeptide, D-alanyl D-alanine is added to form the pentapeptide side chain. The NAM pentapeptide is transferred from UDP to bactoprenol phosphate at the membrane surface. Then NAG is transferred from UDP NAG to the bactoprenol NAM pentapeptide to form the final disaccharide pentapeptide precursor. The completed NAM-NAG precursor is transported across the membrane to its outer surface by the bactoprenol carrier lipid. The disaccharide pentapeptide is then transferred to the growing end of the peptidoglycan chain to lengthen it by one repeat unit. As the bactoprenol pyrophosphate returns to the inside of the cell membrane, one phosphate is removed by a pyrophosphatase, leaving a bactoprenol phosphate to accept another NAM pentapeptide. Finally, the peptide side chains are linked together by transpeptidation reactions, forming peptide bonds. In E. coli, the free amino group of diaminopomelic acid is linked to the carboxyl group of the subterminal D-alanine, releasing the terminal D-alanine. Other enzymes remove the terminal D-alanine from side chains that are not cross-linked.
The gram-positive cell wall is thick. It's peptidoglycan. It's about around 60 nanometers on average, but it also contains something called teichoic and lipoteichoic acid that help maintain the cell wall and help it enlarge. It also gives it a negative charge, okay? So it's going to repel other negatively charged molecules. The gram-negative cell wall is a single sheet of peptidoglycan around 3 nanometers that gives it greater flexibility, but makes it easier to lyse, okay? So the presence of endotoxins, as well as a periplasmic space between the cell wall and the inner membrane, aid in pathogenicity, okay? Degradative enzymes can live here that break down drugs that are trying to kill the cell. And the result is it's easier for them to cause disease, right? Those special spaces in a gram-negative, there's 2 of them, right? Whereas in a gram-positive, there's just the 1, okay? So those special spaces are where shenanigans, bacterial shenanigans can go on, right? Where we can basically knock out antimicrobials and render them harmless, okay? So gram-negatives, think pink, often are better at causing disease. Now, we talked a little bit earlier about the gram staining procedure as a way to determine the type of cell wall. And what you're looking at here is just a diagram of how the gram staining procedure works, okay? And what I would like to do is sort of show this to you. You'll see it again, but I want to show this to you in a video that really outlines it quite well. So let's do that. Let's go get gram stain. Oh, I didn't want to. Well, I didn't want that, I guess. Oops, here.
The gram stain is
So let's hop over here.
method in microbiology. It is a differential stain because it divides bacteria into 2 classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of Graham's iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick, dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
Okay. So back to where we were. You can see that the colors of the cells are going to change as we go through each of the steps. And so that's one of the reasons why when you do a gram stain, you have to make sure and follow the procedure exactly. Okay. You can over decolorize and make everything turn pink, or you can under decolorize and everything stays purple. Okay. So those steps are critical. And it's also important to make sure that when you make the slide that you have an appropriate density of the cells on the slide. Okay. And if we were in an actual lab, I'd show you how to do that, but there are simulations that are on Connect that'll help you out with that. Okay. And there are other types of cell walls as well, right? Acid-fast bacteria include things like mycobacteria and Nocardia, and they contain mycolic acid, which is a long chain fatty acid. Acid-fast bacteria use this to increase their pathogenicity because of its resistance to certain chemicals and dyes. Okay, it's highly hydrophobic. Okay, and in addition, it's going to bear a negative charge. All right, so it's going to repel a lot of different stuff. Okay. Archaea, which are extremophiles. Let's write that down. Extremophiles. TR there. Extremophiles means they like extreme conditions. Like extreme pHs, extreme temperatures, extreme pressures. That's their deal. Okay. They have cell walls that are made up of polysaccharide, some of protein, and all lack peptidoglycan. Peptidoglycan is something unique to bacteria, right? Now, archaea are prokaryotic, but they are not bacteria. Some don't even have a cell wall, right? And these were probably the first cells to show up on the planet, the archaea, okay? And they're still here today, but very few of them cause disease in humans, okay? Mycoplasms naturally lack a cell wall. They contain sterols. Hold on one moment. So they naturally lack a cell wall, okay, that stabilizes the cell against lysis, is responsible for conditions such as walking pneumonia, which would be basically an inflammatory process that can move throughout the body, okay? The L forms are bacteria that normally have a cell wall but lose it during a portion of their cycle. And the result of that is that certain antibiotics are not effective against those bacteria, okay? And the result is that they can persist through that treatment, right? L forms lack the cell wall. The structures that are targeted by antibiotics include the assembly of peptidoglycan, the assembly, in some cases, of lipoteichoic acid. Drugs that attack cell wall synthesis include penicillins and their relatives, cephalosporin, and carbapenems, okay? The gram-negative outer membrane is a lot like the plasma membrane, except that it's going to have different membrane proteins embedded in it, okay? Lipopolysaccharide is a feature of this layer, okay? And can act both as a signaling molecule, but can also act as an endotoxin, okay? Now you might say, well, what is an endotoxin, okay? An endotoxin is a toxin embedded in the cell, okay? So it's tucked in there, right? Exotoxins are secreted. Bacteria do both, okay? Porins are membrane channels that only allow certain chemicals to penetrate, right? So that it's difficult for just any drug to get through there, right? So the cytoplasmic membrane, which is common to all cells, is a phospholipid bilayer with embedded proteins that can either be integral, which means they span the bilayer, or peripheral, which means they're on one or other side, okay? Why is the plasma membrane important? Because it's a semi-permeable barrier, number one. Number two, it keeps the cytoplasmic components in one place, okay? And number three, okay, it can tell the cell what's going on around it, all right? So a lot of stuff goes on at the plasma membrane. It regulates the transport of nutrients and waste because it is semi-permeable, right? The phospholipid bilayer only will permit lipid-soluble materials to pass through it without help. Anything that's charged or polar is locked out unless it has a special transport protein for it embedded in the membrane, right? So that is the unique quality of the plasma membrane that allows the cell to remain functional. It also features something called membrane potential, all right, which allows for certain types of transport to take place. In the case of bacterial cells, the membrane potential allows for energy production, okay? And membrane potential is just a separation of charge across the membrane, okay? The outer membrane of gram-negative bacteria has an extra barrier that is difficult for certain antimicrobials to penetrate, so it's tougher to kill than gram-positives. Alcohol compounds dissolve lipids in the outer membrane, and that's why they're used to clean the skin before certain medical procedures, okay? Treatment of infections caused by gram-negatives requires drugs that can cross that outer membrane and get in the cell and do damage, okay? The cytoplasm is mostly water, but also contains proteins, sugars, waste products, electrolytes, okay, carbohydrates, salts, okay, and it's basically a pool of building blocks for synthesis and for energy, okay. The bacterial chromosome and the extra chromosomal DNA, such as plasmids, contain information for building certain proteins that provide special characteristics for the cell, okay. The chromosome is contained in something called a nucleoid in the bacterial cell, which is attached to a membrane protein that is basically embedded in the cytoplasmic membrane, all right? But as we stated before, there's no nuclear membrane here, all right? So it's called a nucleoid instead of a nucleus. And then the plasmids are outside of the bacterial chromosome. They're shaped like a ring, okay? And they can provide traits such as drug resistance or toxin production, okay? And they can be transferred from one cell to the other using a pilus, okay? Ribosomes are where protein synthesis takes place, all right? What the ribosome does is it binds to a molecule called the messenger RNA, mRNA. Okay. Ribosome binds. Okay. In addition, amino acyl tRNA is brought to the site, okay? And translation occurs. What is translation? That's the building of a protein using the instructions contained in a messenger RNA, which is basically a copy of a gene. That process is known as transcription. Okay. So gene in the DNA undergoes transcription. The messenger RNA made from it is like a temporary copy, and that's what's used for translation. Okay. In prokaryotes, transcription and translation happen in the cytoplasm simultaneously, okay, at the same time. In eukaryotic cells, transcription happens in the nucleus and translation happens in the cytoplasm, okay. The ribosome is made up of protein and ribosomal RNA, which is like the glue that holds the proteins together. There's a small and a large subunit that have certain sedimentation characteristics. We call the small subunit the 30 and the large subunit the 50S. Together, they make something called the 70S ribosome, okay, which is prokaryotic, meaning only prokaryotes have this kind of ribosome, right? The ribosomes in our cells are what are called eukaryotic ribosomes, and they are structurally different but functionally identical. Eukaryotic, eukaryotic, right? You might think, well, the math doesn't add up here. 30 and 50 is 80, right? Well, this isn't a mass. This is a sedimentation coefficient. And this is how fast the thing gets to the bottom of a centrifuge tube when it's being spun. All right. And that has to do not just with mass, but with shape. Right. So that's why you can't just add these up together and get the total S designation. Right. You can see what the subunits look like here. Looks like kind of like a hamburger bun. Okay. Now what goes here? Well, what goes here are transfer RNA. Okay, these sites are called the E, the P, and the A site, right? What goes here is the amino acyl peptide. Okay. And what goes here is the amino acyl transfer RNA. And the direction of travel is this way, from what's called the 5' or the start of the message to the 3' or the end of the message. Okay. We'll learn more about translation in a bit. Okay. Inclusion bodies are used for storage of something. Okay, could be food, could be pigment, could be waste products, can sometimes be gas. All right. All sorts of things can be stuck in these inclusion bodies. Bacterial microcompartments are made of protein and have usually a geometric shape, and they have enzymes that work in particular biochemical pathways. So they're sort of like a proto organelle. Okay. Almost a membranous organelle, but they don't have a membrane. Okay. So a proto organelle. The cytoskeleton is going to be the mechanism that provides itself shape. Okay. Contains actin-like protein. It can be arranged in a helical shape and provide support for the cell. We also found this in archaea. Okay. This is unique to non-eukaryotes and a target maybe for antibiotics, right? And when you make a good antibiotic, it has to be going after something that only the pathogen has, right? Endospores are made by Clostridia, Bacillus, and Sporacina, and they are the bacterial equivalent of seeds, okay? The vegetative cell is the part that's growing and moving and doing all the work. Well, the spore, the endospore, is the temporary site where the genetic information and some cytoplasmic components are going to be stuck until environmental conditions get better. Okay, so not every bacteria can do spores, but a lot can, and a lot that make diseases can, right? Endospores and cysts can be destroyed using a process called autoclaving, which utilizes pressures of 15 psi and temperatures of 121 centigrade for a minimum of 15 minutes. Okay, so let's take a look at spores.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
So the idea behind the endospore is to survive difficult environmental conditions, right? Examples: Bacillus anthracis is the agent of anthrax. Clostridium tetani causes tetanus. Clostridium perfringens, gas gangrene. Botulinum, botulism. C. diff, GI disease, right? And all of these can sporulate, which means that they're much more difficult to get rid of, okay? Spores, cysts, seeds are all examples of mechanisms used by organisms to survive difficult conditions, right? They're sort of like a way to go into suspended animation until things get good again, okay? All right. The archaea are prokaryotic, but more closely related to eukaryotes than they are to bacteria, okay? And we know that by comparing their genetic information, okay? They have a unique sequence of ribosomal RNA, have a novel method of storing DNA, and have novel outer layers, including membrane lipids, cell wall components, and pillin proteins. And as we said before, they like extreme conditions, extreme pHs, extreme temperatures, extreme salt concentrations, extreme pressures, okay? They do it all, right? Different archaea for different parts. Okay, hold on. Some archaea can cause disease, but you're very rarely going to see these. Now, why can these organisms live in these extreme conditions? And that's because they have the appropriate information in their DNA to build the proteins that let that happen. All right. They actually don't like, most of the time, what we would think of as body conditions. Okay. Human body conditions. So you can see here a comparison of the different types of microbes, right? Bacteria, archaea, eukarya. Okay. This is in your book, table 3.2. All right. Now, one of the challenges that we have when we're dealing with bacteria is that we have to figure out a way to understand what we're dealing with in, say, a particular case for a patient or in a particular environmental sample by using information that tells us what category we want to put that particular microbe in. So that could be its shape, that could be its ability to move, that could be what sort of media it's able to live on, okay? All sorts of different things that we can determine, all right? And we can use a reference called systemic bacteriology to strictly use the information in the bacterial DNA to figure out what particular organism we're working with, or we can use the old-fashioned method, which is determinative bacteriology, which uses the traits that I was talking about, right? Phenotypic traits, meaning physical traits, right? And those are the kinds that we generally teach in lab on campus, all right? And that's because we don't have a machine to do DNA analysis, okay? Although it's not terribly difficult to do. Hold on just one moment. So when we do determinative bacteriology, we use techniques such as growth on different media, um, the shape and the arrangement of the cells when we look at it under a microscope slide, and the ability to produce particular biochemical products, right, as a way to determine what we're dealing with, right? So there's 4 major divisions of bacteria and archaea. Versiculocutes are gram-negative bacteria with thin walls. Firmicutes are gram-positive with thick walls. Tenericutes, bacteria that don't have a cell wall, and mendocicutes are the archaea, okay? Bacterial species are collections of cells, all of which have a particular pattern of traits and share at least 70 to 80% of their genes, while a subspecies have slightly different characteristics, okay, but are still basically a member of that umbrella, that bacterial species, right? And a serotype is an even smaller category representing a bacteria or an archaea with a particular surface molecule that we can detect using things like antibodies that are attached to dyes or attached to enzymes, okay? So these are all different ways to characterize. When we characterize a serotype, okay, let's be specific here. When we do a serotype, we have to use techniques like ELISA, which is enzyme-linked immunoabsorbent assay, okay, or we use special type of microscopy called 2-photon microscopy, okay, in which we shoot a laser at a slide that has been treated with an antibody that has a dye attached to it that glows when the laser hits it, okay? So that's very specific, right? Here we use biochemical and morphological clues, okay? And that is the same for the bacterial species as well, right? So this goes for both of these characteristic categories, all right? Okay, we will break here and resume at 3 o'clock, all right? And we will take up chapter 4. So I will see you guys at 3. All right. Now let's head for chapter 4. Okay. All right. I'm going to talk a little bit about the eukaryotic cell now. Okay, the eukaryotic cell came after the prokaryotic cell and is more complex. Okay, first appeared around 2 to 3 billion years ago and came from the same common ancestor as the bacteria and the archaea, right? but was an elaboration on the prokaryotic architecture. Okay. The first primitive eukaryotes were probably single-celled and independently living. They would probably resemble something that we know today as protozoa. Okay. And we'll get into what protozoa are a little bit later on, but protozoa, protozoa-like. Okay. Over time, the cells formed colonies, and cells within those colonies got specialized for particular functions. Okay. And eventually multicellular organisms evolved from this. Okay. So the result is a more complex body plan. And that's why in multicellular eukaryotes, we have to have things like a circulatory system and a lymphatic system and a nervous system and an endocrine system and so on. Okay. So that's very different from the way it is in a single cell. All right. And you can see down here some different examples of eukaryotic microbes, right? Protozoa are always unicellular. Fungi and algae can be either. And helminths are always multicellular, right? And some helminths you can actually see with the naked eye. So we're going to talk about the eukaryotic cell, okay? The ins and outs. You can see an example of the eukaryotic cell here, and we're going to go into what each part does. Right? You've got the glycocalyx. You may or may not have a cell wall, depending on which kind of eukaryote we're talking about. You'll always have a plasma membrane, indicating that here. Yeah, cell membrane. So, yeah. Shame on that. They should indicate the plasma membrane. It's right there. Okay. Inside you have the cytoplasm, and then you've got features such as membranous and non-membranous organelles, right? Membranous organelles be things like mitochondria, chloroplasts, and Golgi body. Non-membranous would be things like centrioles and ribosomes and cytoskeletal components, okay? And we'll get into what each one of them does. But the other thing you should notice about it is it's much larger than a prokaryotic cell. Okay? So generally, you can see eukaryotic cells on sometimes on scanning, on the microscope, which is 40x total mag or low power, right? Whereas in bacteria, you always need the oil immersion. And you might notice something else. This is relative size scale accurate, right? Notice that the bacterial cell is about the same size as a membranous organelle called the mitochondrion, or the membranous organelle called the chloroplast, right? And there's a reason for that, which is that they are thought to have descended from bacteria, okay? Eukaryotic flagella are functionally identical, but structurally different from bacterial flagella. They're thicker, they're more complex. They're made up of microtubules. They use ATP for power, okay? Eukaryotic cilia are a lot like flagella, except that they're shorter and more numerous, and they're only found in a single group of protozoa and in certain animal cells, like in our body. They're found in the oviducts and the vas deferens and in the upper respiratory, okay? The glycocalyx is, again, an outermost sugar layer made up of polysaccharide that can either be a slime layer, a capsule, or just a network of fibers. And it allows for signal reception, attachment, and it also helps protect the cell because it's another layer, okay? Now, Cell walls are something that some eukaryotes have and some don't. Okay, all plants have cell walls. Okay, fungi have cell walls. Okay, fungi are microbes that live off of decaying material, dead material. Okay, protozoa and helminths don't have cell walls. Okay, they are animals. Cell walls of fungi provide support. They're different chemically from prokaryotic walls or from plant cell walls. The thick layer of polysaccharide is composed of chitin, or in some cases of cellulose. Okay. And the thin outer layer is mixed glycan. So you can see an example here. Okay. The cell membrane is a phospholipid bilayer with embedded proteins, which again acts as a semi-permeable barrier. All right. Keeps the internal contents in one place. There are sterols embedded in the phospholipid bilayer that allow for a reduction in the mobility of the phospholipids. Okay. And it is also important in transport and in signal reception, right? It tells the cell what's going on outside the cell. The nucleus is a prominent organelle in the eukaryotic cell. Okay. It's found in the cytoplasm inside the nuclear membrane, which is actually two phospholipid bilayers, one on top of the other, with large pores in the membrane that are formed by integral proteins. And that's so that large structures can go in and out of the nucleus easily, right? The nucleolus is the site of ribosome assembly, and it's also the location of what are called the nucleolar organizing regions, which are the genes that code for the ribosomal RNA. Okay, so the ribosomal RNA gets made there, and then it gets attached to the ribosomal proteins, and then the assembled ribosome leaves the nuclear region through a nuclear pore, and it starts doing its work either on the rough endoplasmic reticulum or in the cytoplasm. The chromatin is DNA and histone proteins and contains all the protein building information that the cell needs to survive. Okay, so it's the blueprint for the tools of life, which is what proteins basically are. Okay, the endoplasmic reticulum is a series of tunnels, membranous tunnels, that transport materials from one location to another. The rough endoplasmic reticulum is where secreted and membrane proteins are manufactured. The ribosomes on the surface extrude the proteins into the internal reticulum, and then it works its way to the end of the reticulum where transport vesicles pick it up and take it to the Golgi apparatus, right? The smooth endoplasmic reticulum is connected to the rough endoplasmic reticulum, but it is for nutrient processing and storage of non-protein macromolecules such as lipids, or in many cases, calcium. Okay. The Golgi is a site of protein modification and shipping. You could think of it as the distribution warehouse for the cell, right? It receives the transport vesicles from the rough ER and then produces secretory vesicles with proteins in the vesicle, all heading for a common destination. And then it drops them off there. Then the vesicle comes back and then the cycle repeats. Okay. They're always closely associated with the ER. The transitional vesicles from the ER are picked up at the face of the Golgi apparatus. The proteins then get modified inside the Golgi by having chemical address labels attached to them. They're made up of polysaccharide and lipid. And then condensing vesicles pinch off the Golgi, and then using the microtubules as a track, they are walked using ATP as a power source to their final destination, where they dump their payload and those proteins start to do their work. Okay. So the nucleus, the ER, and the Golgi are all going to work together to deliver protein to its final destination so it can work, right? A segment of genetic code from the DNA, the gene, is copied into a messenger RNA, and then the messenger RNA passes through a nuclear pore. A ribosome then binds to it, reads it from the beginning to the end, the 5' to the 3' end, and then assembles the protein using that information in a process known as translation. Right? Those proteins in the Golgi apparatus that arrive from the rough ER are chemically modified and packaged into vesicles that are going to eventually arrive at the final destination for that protein to do its work. Okay, so sometimes that will mean becoming part of the plasma membrane or part of a membranous organelle. Sometimes that will mean being extruded from the cell, spit out, basically. And sometimes that will mean becoming a protein inside a vesicle. Okay, it all depends. Lysosomes are like the stomach of the cell, and their purpose is to break down worn out components or food components and then use some of those materials again in biosynthesis, or in the case of waste products, they get dumped outside the cell. Okay. Vacuoles are membrane-bound sacs with fluid or solid particles that are going to be either digested, excreted, or stored. And they're made in phagocytic cells in response to food and other substances that have been engulfed, have been taken in. Contents of a food vacuole are digested through the merger of the vacuole with the lysosome. Okay. If it's solid, okay, it's called a phagosome. Okay. Phagosome merges with lysosome. Okay. Products are broken down. So the lysosome is filled with enzymes that will destroy protein, can tear apart any variety of different polymers. Okay. And as a result, those enzymes need to be kept inside the lysosome for them not to damage the entire cell. And in fact, when the cell decides to check out, i.e. kill itself, one of the things that happens is that the lysosomes basically burp and release their contents and chew up the cell from the inside out. Mitochondria is the powerhouse of the cell. This is where the cell gets the bulk of its ATP. Okay. The mitochondria receives molecules from the cytoplasm like pyruvate and breaks them down into CO2 and water and uses the energy from that breakdown to manufacture ATP, in some cases directly, or in some cases using a device called the electron transport chain, which is in the inner mitochondrial membrane. The mitochondria have two membranes around them instead of just one. Same with chloroplasts. Okay. Mitochondria also have their own DNA, and mitochondria have their own ribosomes. And the DNA and the ribosomes are very similar to bacterial DNA and ribosomes. And that gives rise to this idea of endosymbiosis, which we'll get into in just a bit. The chloroplasts are found in anything that does photosynthesis, algae and plant cells. Okay. They can convert energy from sunlight into sugars, and then those sugars can be oxidized, and that provides ATP for the cell. Okay. One of the byproducts of photosynthesis is oxygen, which is a critical component of respiration, right? Respiration uses oxygen in order to break down glucose. Okay. The chloroplast looks like mitochondria, but it's a bit bigger. There are pigments in it that are able to capture light energy and funnel that into ATP synthesis, and they have a variety of shapes. Okay? So this is a look at this idea of endosymbiosis.
The eukaryotic internal membrane system, called the endoplasmic reticulum, and the nuclear envelope may have evolved from infoldings of the plasma membrane in an ancestral prokaryotic cell. Such infoldings are common in modern prokaryotic cells. The theory of endosymbiosis proposes that a critical stage in the evolution of eukaryotic cells involved endosymbiotic relationships with prokaryotic organisms. Microorganisms that live within other cells and perform specific functions for their host cells are called endosymbionts. According to the theory, energy-producing bacteria may have been engulfed by a larger primitive cell and come to reside within it, eventually evolving into what we now know as mitochondria. Photosynthetic bacteria use photosynthetic pigments embedded in internal membranes to derive energy from sunlight. These bacteria may have come to live within early eukaryotic cells, leading to the evolution of chloroplasts. Several facts provide evidence for the endosymbiotic hypothesis. A few examples include: 1. Mitochondria and chloroplasts contain their own circular DNA, similar to DNA in bacteria. 2. Both mitochondria and chloroplasts are surrounded by two membranes. The inner membrane is probably evolved from the plasma membrane of the engulfed prokaryote, while the outer membrane is probably derived from the cell membrane of the host. 3. Mitochondria are about the same size as bacteria. 4. Mitochondria appear to have been derived from purple bacteria, and chloroplasts derived from photosynthetic bacteria.
Ribosomes are the site of protein synthesis, and they're found scattered throughout the eukaryotic cytoplasm, as well as attached to the rough endoplasmic reticulum. Okay, and their purpose is basically to read messenger RNA and use that information to assemble amino acids into a functioning protein. The information for that is contained in the message, and that's why the process is called translation. You're translating the information from a polynucleotide, messenger RNA, into a protein. Okay. They're also found inside mitochondria and chloroplasts, but those ribosomes are more like prokaryotic ribosomes, right? So we would say again that they are functionally identical but structurally different from eukaryotic ribosomes. Okay. Sometimes they occur in chains on a message, in which case they're called polysomes. So they're making lots of protein in one shot when they're doing that. Large and small subunits of ribonucleoprotein make up the ribosome. The eukaryotic ribosome is an 80S ribosome, which is a combination of a 60 and a 40, right? Remember that the prokaryotic ribosome is a 70S ribosome, right? So it has a different shape and it has different building blocks that make it up, okay? The cytoskeleton provides a framework for the cell, okay? It anchors organelles so that they function effectively with other organelles. It moves organelles from place to place so that they're more efficient, okay? It moves chromosomes during the process of mitosis, okay? It moves RNA and vesicles around the cell using ATP for power, allows the cell to change its shape, and allows the cell to move. Cytoskeletal components include actin, which is part of what are known as microfilaments, microtubules, which are made up of a protein called tubulin, hollow tubes, right? And intermediate filaments, which are rope-like structures, okay? So all of these provide a framework and a transportation system inside the cell and maintain the cell shape, and also provide scaffolding for other proteins to do their work effectively. So sort of like putting enzymes which help chemical reactions speed up in a sort of an assembly line arrangement so that they're more efficient at what they do. Okay. So there's an example of different cell types and the different features. Okay. So this is table 4.2 in your book. All right. Now we're going to talk about fungus. All right. Fungi basically feed off of decaying organic material. Okay. There's 50,000 species of fungi that have been identified up to this point. Macroscopic fungi you can see with your naked eye, and microscopic fungi you have to see through a microscope, right? There are unicellular fungi, there are colonial, and there are multicellular fungi, right? Yeasts are single-celled organisms, have a round to oval shape, can reproduce either asexually through a process known as budding, or they can reproduce sexually using what are called reproductive hyphae. Okay, so they can do either or. All right, hyphae are thread-like cells that anchor the fungus to the thing that it's growing on. All right, some fungal cells are dimorphic. They can take either form depending on growth conditions. Okay, so you can see some examples of hyphae on the right there. In Rhizophus, you've got something called non-septate hyphae. So that's a syncytium. It's a multicellular structure that doesn't have separate cell membranes between the nuclei, right? And then there are septate hyphae, which do. Okay. You can see here sort of the organization of a yeast cell, right? All the components you would expect to see in a eukaryote are there, right? A membrane-bound nucleus, eukaryotic ribosomes, mitochondria, Golgi, lysosomes, and so on, okay? And you can see in this drawing that it's budding, right? It's reproducing asexually, right? And you can see what it looks like under an EM micrograph to the right, right? Remember, though, that yeast can also mate, okay? They can generate these reproductive hyphae that can fuse and produce now a new zygote, right, or spore. And the advantage of that, of course, is that they get greater biodiversity and the cells can resist a variety of changes in the environment, okay? So that's yeast. That's a type of unicellular fungus, right? Other types include the fungi you can see, right? Like if you've ever walked through the woods after a rainstorm and you look at a rotting log, you might see mushrooms sprouting up from the log. And those are examples of macroscopic fungi, okay? So let's take 15 and I will come back at 3:40, okay? And at the back end of this. Home stretch. Now, do fungi cause human disease? Absolutely. Most fungal diseases on humans start with the prefix tinea. Okay, let's write that down. Tinea. Tinea capitis, tinea unguinum. Examples of fungal infections of humans. You can get them in a community setting. You can get them as environmental pathogens, and you can get them as nosocomial infections. Nosocomial means in a clinical setting, right? Harmless spores can also make opportunistic infections in an AIDS patient. Now, what is an opportunistic infection? One that takes advantage of a weak immune system. Okay. Fungal cells give off substances that can cause allergy. Toxins made by poisonous mushrooms can produce death. Those would be exotoxins, right? And aspergillus produces lethal poisons to animals who can eat the contaminated grain, right? Number of species that are pathogenic to corn and grain can cause disease in domesticated animals, cows, pigs, chickens, okay? Fungi can rot fresh produce when they ship, right? You lose about 40% from fungus. But without fungus, we wouldn't have the cycles, right? What do I mean by cycles? We wouldn't have the carbon cycle, the nitrogen cycle, okay? Have the phosphorus cycle. Okay? And to some extent, we wouldn't have the water cycle. Okay? So the fungi are a big part of this. Okay? They can form associations with plants and increase their ability to absorb water and nutrients. And we've engineered them to make all kinds of stuff that we need, such as antibiotics and vitamins, okay? And we use them as food products, okay? Those are different types of fungi. Those tend to be what are called macroscopic fungi. Fungi are heterotrophic, meaning they use an organic source for their energy, okay? Saprobic fungi use nutrients from dying organic material, okay, in order to grow. Parasitic fungi grow on living hosts, but they are utilizing dead material from the living host. Okay, so for instance, in the human diseases like toenail fungus and athlete's foot and ringworm, all right, they're consuming what? They're consuming the dead skin cells. Okay, so it's still dead material. The fungus penetrates the substrate and makes enzymes that reduce it to tiny molecules, and then the cells absorb it. And they thrive when things are nutritionally poor or adverse. They can produce spores, okay? And they can thrive in environments that have a high salt or sugar content, okay? Fungi have a variety of shapes. They can grow as single-celled organisms or as colonies. Colonies of yeasts look a lot like bacteria on media, okay? Colonies of filamentous fungi look fuzzy, okay? The mycelia are the intertwining mass of hyphae that make up the above ground portion of the colony, right? The fuzz that you see growing. Well, the septa are cross walls that are found in fungi that allow materials to flow from one compartment to another, okay? And non-septate hyphae are just one long continuous fusion of cells, while vegetative hyphae are the visible mass that you see, okay? Reproductive hyphae generate spores, okay? And that's a form of sexual reproduction, okay? Spores are one of the ways that the fungi are able to persist through dry or adverse conditions. And they can be dispersed by air and water and living things. They're around us all the time. And if you don't believe me, just take a slice of Wonder Bread and leave it on your kitchen counter for a few days. And what will happen after a time is that you'll see green fuzz growing on it, all right? And that is fungal growth, okay? It can germinate when it finds a favorable substrate, and make a fungal colony in a very short time. Okay. Asexual spore formation, it happens in structures called sporangiospores. You get cell division in a sac-like head called a sporangium that is attached to a stalk called the sporangiophore. Candidiospores are free spores that are not enclosed in a spore-bearing sac. When we have sexual spore formation, we get genes from two parent fungi that produce an offspring with a unique combination of genes that neither parent has, and that provides biodiversity. Okay, put that word in here. Biodiversity increases biodiversity. Okay. Sexual spores vary from a simple fusion of fertile hyphae to a union of female and male structures. Okay, it depends on the fungus that we're talking about. Last eukaryotic, well, second to last eukaryotic group are the protozoa. Okay, the name comes from the Greek for first animal. All right, there's about 65,000 species of these, and they're all over the place. They're in the soil, they're in the water, they're on and in you. Okay, some of them are parasitic and generate human disease annually. Okay, examples that you may have heard of around here: Giardia. Okay, is an example of a protozoal condition. Okay, we can have lepto. Okay, leptospirosis. Okay. Examples of protozoal infections. Okay. Single cell protozoans have all the eukaryotic organelles with the exception of chloroplasts. The cytoplasm has an ectoplasm, which is an outer layer that helps the cell move, and an endoplasm where all of the normal cellular functions go on. Okay. Some organelles are even going to have a primitive nervous system to coordinate their movement. And the cell membrane, of course, as it always does, regulates the movement of material into and out of the organism, right? The cell shape can remain constant or it can change, right? Most protozoans are between 3 and 300 micrometers, which means you could see them with a light microscope, even on low power. Giant amoebas can be between 3 and 4 millimeters, which is quite large, right? Most protozoa are heterotrophic and they need food in an organic form. Many are free-living species that scavenge dead plant or animal material and graze on bacteria and algae. And some have structures like oral grooves that they use to get material inside the cell. Some protozoa absorb food through the cell membrane, such as amoeba, they use phagocytosis. Parasites live off fluids that are in the host. The main limiting factor for them is moisture, right? So you see a lot of these in habitats that are fresh in marine water, contain soil, plants, or animals. They can survive in extremes of temperature and pH by forming what's known as a cyst. Okay. The cyst is the protozoal counterpart to the spore or the endospore or the seed. Okay. When we look at a protozoan, all right, we have a variety of stages in the life cycle. The trophozoite is the feeding stage. It requires food and moisture to stay active, while the cyst is the dormant stage, okay, where conditions are unfavorable, resistant to heat, to drying, to chemicals, and can be dispersed by air currents, and is an important factor in disease spread. You can see an example here of a simple life cycle, right? You've got the free feeding stage, the trophozoite. Then we have poor nutritional conditions. So the result is early cyst wall formation, right? Then we get a mature cyst. the resting stage. This is resistant to a nutrient poor environment. Sorry. Environment. There we go. Okay. Then the environment improves, the cysts break open, and the trophozoite reemerges. Okay. Some protozoans are only trophozoites, and some alternate between trophozoite and cyst. Some examples that cause disease: Trichomonas vaginalis, which is an STD, doesn't form cysts and is transmitted by contact with infected bodily fluids. Entamoeba histolytica and Giardia lamblia form cysts that are already transmitted and contaminated water and food. All protozoa reproduce by mitosis, okay, or by multiple fission. Sexual reproduction can occur in protozoa. Ciliates participate in conjugation with fusion of cells and exchanging of micronuclei, resulting in, again, an organism with a unique combination of genetic characteristics. So you can see here some examples of the different protozoa, okay? This is in your book, table 4.4, okay? Now we move to the helminths, okay? These are worms. This is a more complex organism. It's the first one, well, aside from the fungi, it is multicellular. Multicellular and eukaryotic. Okay. That's characteristic shared by some fungi as well, right? These include tapeworms, flukes, and roundworms. Adult specimens are big enough to be seen with the naked eye. Some flatworms and roundworms are parasites, but a lot live free in soil and water. Parasitic helminths spend some of their lives in the GI tract of the host. Flatworms or nematodes have a thin, segmented body plan and can be divided into the cestodes, which are tapeworms, and the nematodes, which are flukes. The roundworms have an elongated, cylindrical, unsegmented body. Okay, so you can see some examples of these here, right? Notice here we have a segmented body plan. Segmented, which is more complex. Okay, you'll also notice that there are body systems present here. Okay, so there's a nervous system, there's a circulatory system, and so on, right? The most elaborate body systems in these organisms are the reproductive and the GI, right? That's because it relies on the host for everything else, okay? Multicellular animals equipped with organs and organ systems make this the most complex of the pathogens we've dealt with. The most developed organs, as we indicated, are the reproductive and the GI. As a result, there's a reduction in other systems, okay? They have a complete life cycle that includes a fertilized egg, a larval, and an adult stage. Adults determine, or adults get their nutrients and reproduce sexually in the host. In nematodes, the sexes are separate and different in appearance. In trematodes, the sexes are either separate or hermaphroditic. What does hermaphroditic mean? It means both primary sex organs in one organism. Meaning what? Well, they have testes and they have ovaries in the same organism, right? Cestodes are generally hermaphroditic, right? The life cycle is to transmit the infective form, which can either be an egg or a larva, to the body of the host. The host in which the larva develops is the intermediate host. Adulthood and mating occur in the definitive host. Okay. Sometimes the intermediate and the definitive host are the same. Sometimes they are different. Hold on. So sometimes the definitive and the intermediate host are the same. Sometimes they're different. Okay. Sources of human infection include contaminated food, soil, and water. Routes of infection, oral intake, or penetration of either broken or unbroken skin, right? Some of these are nasty enough that they can go through intact skin. Example would be hookworm, okay? Fertilized eggs get released into the environment. They have a protective shell and food so they can develop into larvae, but they are vulnerable to heat, cold, drying, and predation. Some helminths can lay almost 25 million eggs a day to make sure that the life cycle gets complete. That's a lot of eggs, okay? So you can see some examples of the different types of nematodes, of worms, of cestodes, trematodes, nematodes, okay? This is table 4.5 in your book. If we take a look at one example of a life cycle, okay, we can see the pinworm, right? Pinworm formal name is Enterobius vermicularis. Common infestation of the large intestine, can be up to 12 centimeters long with a tapered, curved cylindrical shape. The eggs are swallowed, so they get picked up from another infected person or objects that they've touched. The eggs hatch in the intestine, the larvae mature within a month, and the male and the female worms mate. And you can see in the inset below that they look different, okay? The female then migrates to the anus to deposit the eggs, which is followed by itching, okay? The scratching is then going to spread the eggs. This is one of the ways you can tell if you've got a worm infection is to put tape over the anus. And then if you look at the tape, the eggs will be attached to it, okay? You can do the same for your pets, right? That can get worms. There are about 50 species of helminths that hit humans, and they're distributed in all areas of the world. There's usually a higher incidence in tropical areas. Yearly estimates of cases in the billions are not confined to developing countries, okay? Some estimates include 50 million infections in North America alone, okay? So they're all over the place, right? Tapeworm is probably the one that you're most familiar with, and there's a little video on tapeworm that I'll show you in just a bit here. So let's walk through some of the stuff we've discussed today. So let me round up some stuff for you. Yes. So, let's see. Got this. I've got, there we go. Here's the MP4. Okay. That's good. Let's see. Share.
Cells actually carry out life functions.
It contains the information, unit of life.
Everything that is living is composed of either one cell, whether it be bacteria or protists, or multicellular.
Everything from humans to oak trees to fish and bacteria have cells.
The cell, the basic unit of life. The cell is the simplest component of living matter that can carry out all of the activities necessary for life in every organism. The simplest life forms are made up of only one cell, and in fact, the vast majority of organisms are single-celled, like bacteria, amoebas, and paramecium. More complex life, like plants and animals, are multicellular, and these organisms can have trillions of cells. No one really knows the exact number of cells in the human body. It is estimated that there are anywhere from 10 trillion to 100 trillion cells, and about 200 different types of cells, with different shapes and various functions. While there is no such thing as a typical cell, most cells have chemical and structural features in common. The individual cell is actually a system unto itself, comprised of atoms and molecules, which combine to form proteins and various organelles which structure the cell. In multicellular organisms, like cells group together to form tissue, and tissues in turn combine to form organs, such as the heart. But the heart has many different types of cells to help it function. There are red blood cells which transport oxygen, white blood cells which help fight disease, and cardiac muscle cells that help the heart contract and pump blood throughout the body. Organs function together to make up organ systems, which ultimately make up an organism. And each cell's activity is dependent on the subcellular structures within the cell.
So a cell is not just something that we look at under a microscope and see, oh yeah, there's a cell. The cell is an activity, just as if you were to take a human city and speed it up tremendously with the way that all the people are moving around. So there's all this motion going on inside the cell.
The key to understanding how the body works, or how plants make food, as well as understanding the new frontiers like biotechnology and genetic engineering, is dependent on understanding cells. Most cells are microscopic. They cannot be seen without the aid of a microscope. But if you look at a slice of an orange, you will see it is actually made up of lots of tiny sacs. These sacs are large plant cells, visible to the naked eye. Cells are measured in micrometers and nanometers. A micrometer is 1/1000th of a meter, and a nanometer is 1/1000th of a micrometer. Because cells are so small, they remained hidden and unknown for centuries. It wasn't until the invention and development of microscopes that this miniature world was finally revealed, allowing scientists to study and begin to understand cellular life. In 1839, two German biologists, Matthias Schleiden and Theodor Schwann, developed an idea called cell theory. While looking at thin slices of animals and plants through a microscope, they saw how everything seemed to be made of tiny chambers. They called these chambers cells. Their theory states that all living organisms are composed of cells, and that cells are the building blocks of life. And while these were important revelations and are still accepted today, they still didn't know where cells came from. In 1850, Rudolf Virchow saw something that would change this. In his microscope, he observed cells pinching in two. He realized that cells actually come from pre-existing cells through cell division, and added this to the cell theory. Over the years, scientists have continued to develop this theory, and modern cell theory now states that all organisms are made up of cells, that new cells are always produced from pre-existing cells. The cell is a structural and functional unit of all living things. The cell contains hereditary information, which is passed on from cell to cell during cell division. And all cells are basically the same in chemical composition and metabolic activities. Cell biologists still cannot describe all the details of how cells produce the quality we call life. But recent research in genetics, developmental cell biology, immunology, and neurobiology have led to great strides in understanding structures, functions, and evolution of the cell. There are different types of cells, and certain cells have different functions, yet there are things all cells have in common, and one is a cell membrane that surrounds and encases the cell.
The cell has a membrane around it, which has various protein molecules on that membrane that serve as gates to let certain chemicals in and out of the cell. So the membrane is very important, kind of like the border of a nation is, with its gates to let things, people or goods in or out of the nation. That's the membrane of a cell.
It determines what substances are going to pass right through. It determines what substances need to have a messenger to take them through. It maintains the integrity of the cell. It contains all this material, all this organic material, the unit of life, from the outside world.
Within the cell membrane, there is a jelly-like substance called cytoplasm, which aids the structure and function of the cell. And while all cells function similarly, there are some important structural differences. Cell biologists group cells into two categories: prokaryotic and eukaryotic. Bacteria and archaea are prokaryotic cells. These represent the simplest, most primitive life on Earth, as prokaryotic cells are more simple than eukaryotic cells.
And that means that they don't really have a nucleus and they don't have a membrane that encloses the organelle. So everything is kind of, dispersed in the fluid of the cell, in the cytoplasm.
All other organisms like protists, fungi, plants, and animals are made up of eukaryotic cells, which are more modern and complex.
And one can even talk about the evolution of cells, the way cells have gotten more complex over time. Basically, our human cells are more complex than bacteria cells.
Eukaryotic cell has a true nucleus where the DNA is encased instead of floating around in the cytoplasm, and the rest of the organelles are encased by a membrane as well.
Organelles are special membrane-bound structures inside the cell which carry out specialized functions.
Just as we have organs within our body, the cell has organs within it. Because the cell is very small, these organs are called by cell biologists organelles. The animal cell has a very important organelle called the nucleus, which is where the DNA is housed.
DNA, deoxyribonucleic acid, is found in all cells. It is composed of molecules which carry genetic information. It is, in a sense, the instruction manual or blueprint for cells that specifies how proteins are assembled to regulate the cell.
What is it that DNA does? Is it the boss in the cell? Is it the kind of government in the cell? Not really. I look upon it more like the U.S. Constitution, in that it has to be actively gone to by the society itself and used to structure the government. And that's for our society. For a cell, the DNA as a code is gone to by all the other molecules of the cell that are spinning around in their various interactions and metabolisms that are going to the DNA for the pattern of the code that makes the amino acids, that makes the proteins.
Chemical reactions and enzymes produce proteins which carry out the cell's activities, and ribosomes are where the proteins are made.
There are the ribosomes, small little bodies, and very, very many of them inside animal cells. And in fact, ribosomes are in all cells. And that is where the amino acids, small little building block molecules that make proteins, are brought together into chains, and these chains fold up into the proteins that are used, that all cells need. And that occurs on the ribosomes of the cell.
There's the endoplasmic reticulum, or ER, which is a network of membranes that encircles the nucleus and extends into the cytoplasm. The ER is responsible for synthesizing and assembling proteins and is where the ribosomes are located. There's the Golgi complex, a group of flattened sacs that are responsible for processing, sorting, and modifying the proteins. And the lysosomes, which are small sacs of digestive enzymes that break down ingested materials, secretions, and wastes. They are, in a sense, the stomach of the cell. Then there are the organelles called mitochondria. They are smaller than the nucleus, and there tend to be many of them throughout the cell.
And these mitochondria are the power plants of the cell. They convert the food that comes into the cell and combine it with oxygen to create energy molecules that power reactions in the rest of the cell.
Though plant cells are eukaryotic and have many of the same organelles as animal cells, such as a nucleus and endoplasmic reticulum, the plant cells have certain characteristics which are significantly different.
When we look at the plant cells, we see major differences with animal cells. Plants, of course, don't have skeletons. Each cell has its own skeleton, and that's the cell wall that's outside of the cell membrane. These cell walls are economically quite important. For instance, wood is all cell wall. Cotton fiber is a very pure kind of cell wall made up almost exclusively of cellulose.
But one of the most important differences distinguishing plants from animals can be found inside the cell. Plant cells have chloroplasts, organelles that contain a green pigment called chlorophyll, which traps light energy from the sun and combined with water and CO2 makes food for the plant through a process called photosynthesis.
And then there's one other structure that's extremely important that is largely unique to plant cells, and that's the vacuole. The vacuole is where water is stored. This ability to store water, to regulate water, is one of the things that makes plant cells be able to respond so rapidly to the environment. Very quick motions are centered around changing water distribution in plants.
As cell theory states, all cells come from previously existing cells. This happens through cell division. Cell division, called mitosis, is an amazing and quite elegant process that enables organisms to grow and reproduce. And though there are some differences, This process is remarkably similar in all organisms. Through a sequence of steps, replicated genetic material of a parent cell is equally distributed to 2 daughter cells. It starts as all the chromosomes or genetic material are organized into entangled structures called chromatin. In the first phase, called prophase, the chromatin condenses into distinct chromosomes, and spindles that provide the mechanics for the division form at opposite poles of the cell. In the second phase, called metaphase, the chromosomes are aligned, equally distant from the 2 spindled poles. Next, in the anaphase, the paired chromosomes move to opposite ends of the cell. In the last phase, telophase, the chromosomes are cordoned off in distinct new nucleuses. This happens as cytokinesis divides the cytoplasm, making 2 distinct cells. Understanding the basics of cell structure and function is the foundation for understanding the future of science. Cell biologists, geneticists, and other scientists are investigating the properties of cells to find new ways to manipulate and replicate cells for use in agriculture, such as producing healthier and hardier plants. And in medicine, stem cell research is making strides in showing scientists how to make cells differentiate to do specific jobs, like increase insulin in diabetics, or possibly find cures for diseases such as Parkinson's or multiple sclerosis. Further investigation into how cells evolved and are continuing to evolve may uncover many of the yet unsolved mysteries of our world.
Okay, hold on.
We've spent the past few months talking about animals here on Crash Course, specifically human animals, because humans, we love talking about ourselves, and also because animals are just really interesting. But it's high time that we talked about the rest of the living world, because I hate to break it to you, but most of the alive things on Earth are single-celled organisms. And by most of the alive things, I mean that these organisms make up 2 of the 3 taxonomic domains of all life, plus 1 of the 4 kingdoms. I'm talking about archaea, bacteria, and protists. With the exception of a few protists, they're all unicellular, and they are by far the most abundant and diverse organisms on Earth. Maybe more importantly, they lay claim to the world's oldest and earliest living lineages, dating back to the very first twinkle of life on this planet. So by understanding these 3 groups, you begin to truly understand life on Earth, its origins, and how everything that came after them, including us, came to be. What's more, because their heritage is so ancient, these organisms often take weird, cool forms that don't look like life as we think about it, and they do amazing things. Some not only live but thrive in environments that would kill you, me, and everything we hold dear, and others make their living by invading organisms, including us, and causing disease. And there are those that do the opposite, making life possible by fixing nitrogen from the atmosphere, helping animals digest food. Members of these groups have names like sailor's eyeballs and dog vomit slime mold, and they can take the shape of rods, blobs, corkscrews, or coils. Kind of like the doddering, eccentric relatives you're forced to spend some holiday with once a year, the archaea, bacteria, and protists are our oldest,
oddest relatives.
And it's about time you got to know them. There's no denying it. Every multicellular organism on this planet, whether it be a mushroom or a vampire bat, evolved from a single-celled organism. And while some of these single-celled organisms evolved to populate the world as rhinos and strangler figs, others found happiness in a unicellular lifestyle, and they haven't changed much in the past few billion years. Today, nearly all unicellular organisms are either archaea, bacteria, or protists. Protists, you'll recall, are eukaryotic organisms that make up the kingdom Protista under the domain Eukarya. Bacteria and archaea, meanwhile, are their own prokaryotic domains. And I hope you haven't forgotten this. The big difference between prokaryotes and eukaryotes is that eukaryotic organisms, including you and the plants and fungi and animals that you know, have cells with a nucleus that hold their genetic information, while prokaryotic cells don't have a nucleus or any organelles to speak of. These two groups do have some important things in common, like having plasma membranes that are filled with cytoplasm and ribosomes that contain RNA and synthesize proteins, and they both have DNA that carries the instructions for operating the cell. But eukaryotic DNA comes in strands in the form of chromosomes, while prokaryotic DNA is found in rings called plasmids. So again, and this time with feeling, protists are mostly single-celled eukaryotic organisms. Archaea and bacteria are single-celled prokaryotic organisms. The word prokaryote actually means before the nucleus, which is a clue that prokaryotes are an older form of life. And we literally cannot find anything older than archaea. The first archaea fossils date back to 3.5 billion years ago. I'm talking just a billion years after the Earth formed and was still bombarded by comets and meteors, not to mention fried by UV radiation. But in the midst of all that, archaea or just chillaxing. Earth's climate has calmed down since then, so today archaea are found in some of the world's most extreme environments, in underwater hydrothermal vents, in oil wells, and volcanic hot springs, even acidic mine drainage. Archaea were probably the earliest living things, and their adaptability is probably what allowed them to take root in Earth's early kind of grody environment. One key group of the archaea are the methanogens. These guys perform more moderate environments like mud, swamps, and your intestines, but they derive energy from hydrogen gas and carbon dioxide, which is pretty cool, and they emit methane as their waste product, methanogens, methane generators. We know that waste as swamp gas, and also other kinds of gas. The other groups are extremophiles, which not only tolerate but prefer really wicked surroundings. The most famous of these are the thermophiles, which live in temperatures that would melt your face off. I mean, serious, Pyrolobus fumarii, a species of archaea discovered in the late 1990s in a hydrothermal vent, live at temperatures around 113 degrees Celsius, not Fahrenheit, Celsius, significantly above the boiling point of water. Most organisms can't take heat like that because it causes their DNA to unwind and their proteins to denature or permanently change shape, but thermophiles have evolved adaptations that keep them stable at these screaming hot temperatures. There are also halophiles, or salt lovers, which live in places like the Dead Sea or the Great Salt Lake, probably Daniel Tosh's mouth. Most halophiles breathe oxygen and are heterotrophic, but there are some bizarro outliers, like species that use sunlight to make energy, but not like plants do. They have light-harvesting pigments in their membranes that react with light and enable the cell to make ATP for energy. I know, it's crazy, but despite their alien-sounding ways of life, archaea really aren't all that different from bacteria, which are also prokaryotes. In fact, archaea and bacteria were classified together for much of the 20th century. It was only when scientists realized that they had some important genetic differences, like in the sequence of their ribosomal DNA and the makeup of their RNA, that they were separated into two domains. Bacteria are nearly as ancient as archaea. Fossils show that they were widespread about 1.5 billion years ago, but there's evidence that they've been around for more than 3 billion years. Today, they make up the vast majority of prokaryotes on Earth, and they're super slick when it comes to adapting quickly. Many bacteria are parasitic. Think your strep throat, your staph infection, anything you've ever taken an antibiotic for. But bacteria can fend off antibiotics and the ninjas in your immune system by garbling up their DNA from one generation to another. They can randomly turn genes on and off, creating unique genetic combination as its population multiplies, keeping its host immune system and drug makers on their toes. Like archaea, bacteria don't reproduce sexually, but bacteria have devised a way to pass their genetic material to their buddies, a little trick called horizontal gene transfer. For example, you've heard of antibiotic resistance, right? Well, horizontal gene transfer is one reason for it. A strain of bacteria that has genetic resistance to an antibiotic can pass some of its DNA and that drug resistance to another strain, which is why we're always in kind of an arms race with the bacteria of the world. And of course, bacteria are incredibly diverse with too many phyla to name, more than 24. But one way of classifying them is by their different kinds of cell membranes, which react differently to a staining technique scientists use called Gram staining. Gram-positive bacteria have thick cell membranes, and they're a huge group that includes species that live individually, like Staphylococcus and Streptococcus, as well as some colonial bacteria that are responsible for diseases like leprosy and tuberculosis. And there are lots of gram-negative bacteria too, which have thinner membranes. The biggest group here are the proteobacteria, named after Proteus, because they take so many forms. These include bacteria that make our lives possible by converting nitrogen in the atmosphere to compounds available to plants, as well as others that cause stuff like food poisoning and Legionnaire's disease. Cyanobacteria, meanwhile, are the only prokaryotes that use photosynthesis to make their food, and they're some of the most important members of aquatic food webs, providing microscopic forage for all kinds of freshwater and marine ecosystems. Spirochetes are the corkscrew-shaped bacteria that you've no doubt heard of. Most are harmless, but a couple of parasitic species are the culprits behind illnesses like Lyme disease and syphilis. And speaking of sexually transmitted diseases, the last major group of bacteria worth mentioning are chlamydias, which are strictly parasitic and live only in animal cells. They're scumbags, obviously, and are the leading infectious cause of blindness in the world, as well as that eponymous infection of the urethra that makes me kind of want to cross my legs just thinking about it. So archaea have managed to make a nice multi-billion-year living by surviving in weird, out-of-the-way places, and bacteria have developed ways to pass their DNA without sexual reproduction. But you know who's a hot freaking mess? Protists. Evolutionarily, they're the youngest of the three, having evolved from bacteria around 1.7 billion years ago, and in a lot of ways, they're more sophisticated. For starters, they're eukaryotic, but also some are multicellular, and a few kinds can even reproduce sexually. But their domain is a big crap circus because some protists seem to be more closely related to plants or animals or fungi than other protists, so scientists tend to talk about them based on what else they resemble. There are protozoa, which are kind of animal-like, algae, which are kind of plant-like, and fungus-like ones, including the tastefully named slime molds. The one thing all of these have in common is they need to live somewhere wet, in a bog or in your body or in a snowbank, wherever. Protozoa are actually really cool because they're like tiny animals. Like us, they're heterotrophs, so they have to eat other stuff in order to live. And because they need to eat, they've got like mouth parts, or at least mouth part sorts of things. And they can move around by using all kinds of really cool structures. Some have flagella that look like tails to propel them through the water, or cilia, little hair-like structures that work like oars. And some move around with a kind of blobby amoeba-like motion. I say amoeba-like because the protozoans that move this way are amoebas. And speaking of amoebas, some protozoans are parasitic. You've probably heard of amoebic dysentery. That's caused by amoebas. Malaria is caused by this little guy, a protozoan called Plasmodium vivax, while African sleeping sickness is caused by Trypanosoma brucei, this guy here. Moving on to the plant-like protists, which are algae. All algae photosynthesize like plants, even though they're not plants, because they use different kinds of chlorophyll molecules. Some are unicellular, like tiny diatoms, which have a hard shell made of silica. The amazing thing about single-celled algae is that they can get really honking huge. For example, ladies and gentlemen, cast your gaze upon the sailor's eyeball, thought to be the biggest single-celled organism on the planet, also known as bubble algae. It lives on the seafloor in tropical oceans and can grow up to 5 centimeters across. How is that thing one cell? Anyway, you already know multicellular types of algae, aka seaweed. They're closely related to land plants, as you can tell by looking at them, and they're generally grouped into red, green, and brown varieties, although these all have their unicellular forms as well. The green algae are probably what gave rise to land plants about 475 million years ago. They're the most abundant and diverse, and they have chloroplasts very much like land plants. So they can only live in shallow water because they need a lot of sunlight. Red algae is able to live at greater depths and has an extra pigment in it called phycoerythrin, which gives its chlorophyll a boost in deeper waters. And brown algae is what most of the seaweed you see in the ocean is. Kelp is an example. They're the largest and most complex of the multicellular algae. Finally, we have our fungus-like protists, which include the delightful slime molds. They absorb nutrients from their environment and produce fruiting bodies like fungi. But even though they look like piles of barf, they can actually move around like an amoeba and eat bacteria by phagocytosis. Slime molds can be pretty easy to spot because they're often brightly colored, like this charming species, which in all seriousness is known as dog vomit slime mold. You heard me, these organisms are so freaking screwed up that scientists couldn't think of a better name for it than dog vomit slime mold. Like I said, they're old, they're odd. Get used to it. Thanks for watching this episode of Crash Course Biology. If you want to catch up on anything that you're a little fuzzy on, table of contents over there. Thanks, of course, to all the people who helped put this episode together. And if you have any questions for us, please, Facebook, Twitter, or the comments below. Goodbye.
Classification of phylum Protozoa. B.M. Honigberg classified phylum Protozoa into 4 subphyla. Subphylum 1. Sarcomastigophora. Subphylum 2. Sporozoa. Subphylum 3. Nidospora. Subphylum 4. Xylophora. Subphylum. Sarcomastigophora. Locomotory organs or pseudopodia or flagella. It is divided into 3 superclasses. 1. Superclass Mastigophora. 2. Superclass Opalinaata. 3. Superclass Sarcodina. Mastigophora contains 1 or more whip-like undulating flagella for locomotion. The body is covered by a thin pellicle. Asexual reproduction is by longitudinal binary fission. It is divided into 2 classes. 1. Phytomastigophora and 2. Zoomastigophora. Class 1. Phytomastigophora. It is derived from Greek word phyton, means plant, mastix means whip, and phorus means bearing. As the name indicates, chlorophyll bearing, chloroplasts occur in some euglenoids and in some dinoflagellates. Free-living organisms with autotrophic or heterotrophic mode of nutrition. Example, euglena. Serratium, dinoflagellate with chloroplast. Noctiluca, dinoflagellate without chloroplasts. Zoomastigophora. It is derived from Greek word zoon, means animal, mastix means whip, and phorus means bearing. As the name indicates, chloroplasts are absent. Hence, these are mostly parasitic in nature. Reserve food is glycogen or volutin. Examples, Trichomonas, Trichonympha, Trypanosoma. Superclass 2. Opalinator. Characteristic feature, body is covered by oblique rows of cilia. Binucleate or multinucleate, but all nuclei are identical. Homokaryotic. These are commensals or parasites in the gut of anurans. Asexual reproduction, longitudinal binary fission, and plasmotomy. Sexual reproduction by anisogamy, syngamy with flagellate gametes. Example, Opalina. Superclass 3, Sarcodina. It is derived from Greek word sarcode, means fleshy. Locomotion organelle's body is radially or spherically symmetrical. Some are asymmetrical. Class 1, Rhizophodia. Class 2, Pyroplasmia. Class 3, Actinopodia. Class 1, Rhizopodia. This class includes amoebas, forams, and mycetozoans, slime molds, mostly free-living, few are parasites. Pseudopodia, like Lobopodia, Philopodia, or Reticulopodia are present. Amoeba, Lobopodia, or Philopodia, and foraminiferans, Reticulopodia. In amoebas, body is naked or covered by a test, arcella. Foraminifers have a calcium carbonate shell. Calcareous shells of dead individuals accumulate on the ocean floors and form foraminifera ooze. Examples: amoeba, arcella, alphidium, physarum, and dictyostelium. Class 2, Pyroplasma. Parasites living in the RBC of vertebrates. Locomotory organs are absent. Body is naked and spores are absent. Example: Babesia, which causes red water fever in cattle. Class 3, Actinopedia. This includes Helizoa, Radiolaria, and Acantheria. Marine water forms are Radiolarians and Acantherians. Marine and freshwater forms are Helizoa. Pseudopodia are in the form of Axopodia, Actinopodia. In Radiolarians, internal siliceous shell is present. In Acanthometra, strontium sulfate shell is present. Examples: Calozoum, Radiolarian, Actinophrys, Heliozovan, Acanthometra, Acantherian.
Hello friends, today we are going to be talking about helminths, also commonly known as parasitic worms, are large multicellular organisms which can mature and generally be seen with naked eye. Helminths can be divided into major two groups, platyhelminthes and nemathelminthes. Platyhelminthes, also known as flatworms, nemathelminthes, also known as roundworms. Platyhelminthes haven't body cavity, but nemathelminthes have body cavity. Also, platyhelminthes group divided into major two groups as Cestoda and Trematoda. Cestoda worms, also known as tapeworms, and trematode worms, also known as flatworms. We can see similar segments in cystodonts, but can't see similar segments in trematode worms. Cystodonts have suckers and male and female organ in one organism. Also, megaderms is called as roundworms. Worms are usually caught through table and contaminated soil in warm, humid countries that have poor sanitation and hygiene. If an infected person or animal has detected in soil, having eggs present in their feces contaminate the soil. The egg mature and hatch to produce larvae that grow into adult forms of up to 30 millimeters of length. These adult forms can penetrate human skin, which can happen in person born on contamination soil. The worms then enter the bloodstream and migrate towards the lungs, and also throat, where they are flowed and transferred to the gut. Okay, we'll explain complete details about humans one species by one species in my future videos. Thank you.
Okay. Now, the lab for today, let's go to syllabus. There it is. Lab for today, week 2, is identifying bacteria from gram stain. Okay, so there's a gram stain lab in Connect that should be open at this point. And what I want you to do is to go ahead and run that lab. You'll get those points. I'll enter the points for last week's exercises tonight. All right, so that you get credit for those. And hopefully everybody got their access codes with their books. Okay, they were available with the textbook at the bookstore. If you haven't, please let me know. Okay, so that I can arrange to get you a code. Are there any questions before we bump out? We're good. Okay, I will shoot you this week and last week's podcast so that you can use that to flesh out your notes, and remind you that I've got office hours from 9 to 11 on Mondays. I'll send you that Zoom link. And besides that, I will see everybody in class next Friday. I appreciate you coming.
All right, thank you.
Thank you.